But so we're 2 minutes after, and just in the interest of time, we'll go ahead and get started, and then we'll see if the chat will work. If it doesn't, oh well, next time. All right, so welcome everyone to our fall Global Health Seminar Series. My name is Juliet Sekandi, Assistant Professor here at the University of Georgia, the College of Public Health, the Global Health Institute, and I help coordinate the Global Health Seminar Series that we run through the Global Health Institute for both fall and spring, and that happens every last Thursday of each month. And we've been able to successfully do that over the last 5 or 6 years, I would say. So today we are very privileged to have Dr. Cranmer, who is currently Assistant Professor of Pediatric Infectious Diseases at Emory School of Medicine and Children's Healthcare of Atlanta. She's trained in clinical medicine at Johns Hopkins and in pediatric infectious disease at University of Washington. Her research focuses on tuberculosis detection and prevention among pregnant women living with HIV and their children. She has worked to define the burden of TB infection and disease among children living with HIV in Kenya. And her current NIH-funded project is examining the role of maternal antibodies on infant risk for early TB infection. Dr. Cranmer is also protocol chair for a TB vaccine trial in pre-adolescents in South Africa. I also want to mention that she's also a executive member of the recently funded Emory UGA Tuberculosis Research Advancement Center track, which we are very happy about and very excited about for the next 5 years and maybe more into the future. And so with those a few introductory remarks, I would like to invite Dr. Cranmer to take it away for us. Welcome, Dr. Cranmer.
Thank you so much for the introduction. Can you hear me okay?
I hear you.
Okay, great, perfect. Well, I decided to change the title of my talk just a little bit. So today I wanted to share with you all about pediatric TB vaccination and retitled it Moving Towards the Tipping Point, to just kind of think about where we are in the pediatric TB vaccine field. So I'll first start with discussing the current landscape of pediatric TB vaccines, going from past to present, discussing a bit of the history of TB, then moving on to address the uphill climb of what challenges we have to face for future TB vaccine development, and lastly, tie in COVID to apply lessons learned from the COVID pandemic so that we can get to the final push to get to a tipping point to discovery new and effective TB vaccines in children. So just to give people a little bit of background on the history of TB, TB is an ancient disease. Genetic sequence analysis estimates that the first progenitor of TB co-evolved among hominids found in East Africa. And we found evidence of TB in Egyptian skeletal remains, showing signs of Pott's disease, as you can see in the top picture on the right, around 8000 BC. And as my own research focuses on mothers and children, I find it particularly interesting that the first genetically confirmed cases of TB were among skeletal remains of a mother and infant pair that was found near Israel. The term tuberculosis was first coined by Johann Schulein, also the namesake of vasculitis, Hemlock-Schönlein-Purpura. And on March 24th, 1882, Robert Koch isolated MTB, and we celebrate World TB every year on this day. So the names of tuberculosis also have evolved over time. TB was referenced in Hebrew in the early biblical text of Leviticus, called a wasting disease, and known in Europe as the white plague, for the paleness of a patient's skin, or consumption due to the significant weight loss that patients with TB experienced. So it's a long-standing disease that we've faced. The first efforts to develop a TB vaccine began in 1890 when Robert Koch administered tuberculin, crude protein extract of TB bacillus, as the first therapeutic TB vaccine to be evaluated. Unfortunately, this observational study yielded disappointing outcomes. Among patients with active pulmonary TB disease who received tuberculin, more patients died than were cured. Half of the patients did not have any improvement, and less than 20% improved, similar to the natural history of untreated TB patients. So kind of to go along a little bit of the kind of first half of the 20th century, it brought exciting developments and the worldwide distribution of the BCG vaccine. So M. bovis was first isolated in 1902 by French veterinarian and microbiologist Edmond Nicard. And in 1908, French physician scientist Albert Calmet and his assistant Camille Guerin, veterinarian and prior mentee of Edmond Nicard, were able to subculture M. bovis on this potato bile glycerin medium at the Pasteur Institute in Lille, France. And in 1920, after over 10 years of work involving 230 passages of M. bovis and demonstrating lack of dissemination of their attenuated strain in animal studies, they gave the name Bacille Calmette-Guerin, coined as BCG, for the first TB vaccine designed to prevent TB disease. So today's BCG vaccine, BCG vaccine has widespread coverage and is the number one vaccine used globally, with over 100 million doses every year. And, you know, in the early days of BCG vaccination being available globally, early trial data showed approximately 50% efficacy to prevent TB disease and about 70% efficacy to prevent TB mortality. In these early studies is a meta-analysis showing that. And however, you know, later meta-analyses of studies in the 80s and 90s led to the growing recognition of variability in BCG vaccine efficacy. So ranging very broadly from 0 to 80%. And while I know you can't see all of the fine print of these studies, the number of studies with vaccine efficacy confidence intervals actually crossing 1 is notable. So trials noted variation by age, by latitude, and TB disease type as well. So I added in this paper that just came out, I believe this last month, looking at kind of 21st century, kind of looking at estimates of BCG efficacy. And, you know, this confirmed a lot of our estimates previously, but kind of got a more current estimate of, you know, really what is the vaccine efficacy. And unfortunately, you know, in all ages, vaccine efficacy to prevent all types of TB, both pulmonary and extrapulmonary, was about 20% in all ages, with a kind of higher protective efficacy in young children under 5. Looking at the mortality benefit, all age mortality benefit was about 75%, but this was really mostly seen in the younger population and children. And so this meta-analysis was really helpful to highlight just how much we need a vaccine that has longer duration of efficacy and vaccines to target the older age groups. So just kind of, you know, giving a picture of the global variability of efficacy. You know, one of the earlier meta-analyses did look at like by latitude, and we really see that, you know, there's different higher efficacy in higher latitudes and lower efficacy right around the equator. And, you know, we don't know exactly why this is. You know, is it related to the vaccine strain? Is it related to genetics? Is it related to pre-sensitization with environmental mycobacteria? And this is just a picture of all the different BCG strains genetically, you know, through the worldwide dissemination of BCG in laboratories. There was a series of subculturing so that there was an evolution of strains. And so each strain had, you know, different antigenic, productive, and metabolic properties. And in the 1960s, the WHO, you know, kind of intervened and established production guidelines and primary seed lots of the main vaccine substrains, from which secondary seed lots can be used for commercial production. And today there are 3 central vaccine producers, including Serum Institute of India for the BCG Russia strain, the Japan BCG Lab for the BCG Japan strain, and AJ Vaccines, who makes the BCG Danish strain. It has long been hypothesized that exposure to environmental mycobacteria may affect variability of BCG vaccine response. And while environmental sampling of non-tuberculous mycobacteria in countries with northern and southern latitudes hasn't really actually observed a difference in the prevalence of non-tuberculous mycobacteria in these settings, using environmental sampling studies of soil and water, there is variation of the type of non-tuberculous mycobacteria species by global region. And so the two main hypotheses proposed to explain this variability in BCG efficacy by the non-tuberculous mycobacteria pre-exposure includes the blocking and the masking theories. And this is a very simplified model, but individuals with exposure to non-tuberculous mycobacteria may have these pre-existing immune responses, which blocks the replication of BCG after injection. And then this in turn diminishes the immunogenicity of the vaccine and leading to lower efficacy. And this hypothesis has been demonstrated in a mouse model, and of note, different NTM species had different efficacy levels. Oopsies, sorry. Having a little trouble with my mouse here. There we go. So secondly, the masking hypothesis proposes that efficacy of BCG may be masked by the protection that's already present in the mycobacterial experienced placebo group. So as NTM infection can occur, can offer a degree of protection. And so therefore we see greater gains in efficacy among groups with less sensitization to non-tuberculous mycobacteria and masking the full effect of the BCG vaccine. In addition to challenges in just this variable immunogenicity and variable efficacy of the vaccine, there are safety outcomes to be aware of when using live BCG. In its natural history after BCG administration, the injection site often becomes red and can ulcerate within the first 2 to 4 weeks. And those pictures just show kind of the timeline of what that looks like in terms of day 7 is the first picture, day 14, and then day 21. And then, at around 6 to 10 weeks, it can form a scar, and regional lymphadenopathy can occur about 1 to 3 months after vaccination. More severe BCG complications can include injection site abscess occurring about 25 per 1,000 infants, suppurative lymphadenitis at 2 per 1,000 infants, and while it's very rare, at 3.4 per million, there's disseminated BCG can occur, and that has very high mortality rates. In the pre-ART era, children with HIV had really high rates of disseminated BCG at 992 per 100,000. And WHO at that time made recommendations in 2006 to avoid the use in HIV, in infants with HIV. However, in the ART era, lower rates of suppurative lymphadenitis and no reported cases of disseminated BCG in children with HIV on ART who received BCG. The WHO just recently changed their recommendations in 2018 to actually endorse use of BCG among children living with HIV who had CD4 counts over 200. So this just kind of highlights the overall, last 100 years of the BCG vaccine. I've highlighted a few of these points of, BCG first being administered in 21, but here in 2022, unfortunately, BCG is still the only licensed TB vaccine available. And despite BCG, there's really just continuing high ongoing TB mortality of between 1 and 2 million deaths every year. As a pediatrician, I have to note that 16% of these global TB deaths are among children, and 90% of the TB deaths among children are among children who have not been treated because their TB has not been recognized. This is just a summary of kind of the merits and pitfalls of BCG, which I've summarized a bit of, but it does seem to have just, protection against disseminated TB in infants. And, one of the remarkable aspects of just the immunogenicity is that it does elicit robust CD4 T cell responses, even when given at birth. Many birth vaccines are harder for the little babies to really elicit a good immune response. And as we mentioned, it really is kind of widely administered globally with over 100 million doses every year. But as we've talked about, the efficacy is inconsistent. There are safety risks in immunocompromised, and there's lack of standardized manufacturing procedures has really led to strain variation, and inconsistent supply chain has been an issue. There's been a lot of stockouts, and there's been some neat papers looking at kind of corresponding with those stockouts. Unfortunately, there's just big peaks in TB meningitis for infants, so it really does matter, but the supply chain is an issue. So thinking about the next step after 87 years of BCG as the only available vaccine, the quest for new candidates in children began in 2008 with the introduction of the MVA ID5A trial. And these new trials to look at new candidates have included many different kinds of vaccine candidates, so including viral vector vaccines, protein and adjuvant vaccines, and whole cell vaccines, including both live vaccines as well as killed or inactivated vaccines that are whole cell. When we think about just the vaccine concepts, we kind of tie that to what we're trying to accomplish with the natural history of disease. So, basically there's prevention of infection vaccines. So these are trying to prevent, MTB infection in the first place. And you can see on the left, it kind of just highlights the range of clinical, TB disease. So you can kind of have TB infection, incident and incipient TB, subclinical TB, and clinical TB. And then, kind of after infection, the question is whether the person will actually develop disease or not. And some people are able to control the infection on their own, but some do not. And so that bacterial burden really can continue to rise and then present clinically as TB disease. And so the second, kind of modality of the vaccines are tried to prevent TB disease. So prevention of disease, or POD. After individuals get TB, the WHO does have the aim of trying to get therapeutic vaccines. And so there are studies involving that. And then there also are vaccines that are being tried to prevent recurrence of TB because people who have already had TB in the past have a higher risk of actually having TB recurrence. So for children specifically, the two types of trials that we're employing are really the prevention of infection and prevention of disease trials. Children have not yet been enrolled in any of the therapeutic vaccine or prevention of recurrence trials. And so the target age groups in children really are driven by the epidemiology. So there's a high incidence of TB among infants and among adolescents. And the TB clinical phenotype also does differ by age, with higher lymph node and disseminated disease, highest in infancy, and the corresponding high risk for mortality in that group. And then adolescents really develop more of adult type disease with pulmonary cavitations, which really drives ongoing TB transmission. And modeling studies have shown that a TB vaccine targeted to adolescents has the greatest public health impact, averting significantly more cases per year, even with poor to modest efficacy, which we can see in the red and the blue lines. And this study also showed, in terms of prevention of deaths over a 25-year period, if you have a vaccine with modest efficacy of 60%, it's estimated to prevent 17 million when that vaccine is given to adolescents, compared to under 1 million deaths when that's given to infants. So therefore, there's really been an increased number of trials evaluating new TB vaccine candidates among adolescents. And current infant trials focus on using live whole cell vaccines as a BCG replacement. So I'll go over some of the current candidates that are really being evaluated in trials. So VPM 1002 is a genetically engineered live recombinant BCG vaccine strain. And what this does is it's genetically engineered to replace the urease C gene with listeriolysin. And after the bacillus is phagocytosed, listeriolysin disrupts the phagocytal membrane and allows the vaccine proteins to be released into the cytosol. And this allows for MHC1 presentation to the CD8 cells and enhances inflammasome and autophagy. And preclinical and early clinical trials have demonstrated robust immunogenicity and found that the VPM 1002 had an improved safety profile with higher survival in SCID mice compared to BCG, and no dissemination in other animal models, including guinea pigs and rabbits. The VPM manufacturer, SII, Serum Institute of India, has optimized production steps to enable rapid scalability of the vaccine. for it to move forward if it demonstrates efficacy in phase 3 trials. So the current kind of recently published trial is a phase 2b trial in South African exposed and unexposed newborns. And that demonstrated that VPM 1002 had non-inferior safety compared to BCG. There was a lower grade 3 generalized lymphadenopathy in the VPM arm, and VPM was significantly less reactogenic, so caused less scarring. Less than a quarter of vaccinated subjects had scar compared to 74% of BCG recipients. And children had lower rates of ulceration and abscess formation as well in the VPM arm. However, on the right, when we look at really the multifunctional CD4 and CD8 T cell responses as our markers of immunogenicity, they were lower in the VPM arm compared to BCG. Right now, in terms of the next step, is an efficacy trial. And so it's a multi-center trial in three countries in Africa, evaluating whether VPM compared to BCG will prevent TB infection and disease in infants. And that's a large trial, almost 7,000 infants. And I'll explain a little bit later, but we are doing a phase 1 and 2 trial in pre-adolescents comparing VPM to BCG on children with HIV. The second live vaccine candidate to replace BCG in infant trials is MTB-VAC. And as opposed to BCG, which is attenuated from M. bovis, MTB-VAC is attenuated directly from Mycobacterium tuberculosis. So it retains about 25% of the TB's T cell epitopes that are absent in BCG and preserves antigenic diversity. So to ensure safety, FOP and FAD20D26 genes encoding the TB virulence factors were deleted, and preclinical studies were reassuring for safety in SCID mouse models. MTB-VAC demonstrated lower TB burden in the lungs compared to BCG after MTB challenge for the mice. So a phase 1 dose escalation trial of MTB-VAC versus BCG was conducted among South African infants, and this found no vaccine-related severe adverse events. There was one unrelated death that did occur in the MTB-VAC arm. MTB-VAC had similar reactogenicity profile to BCG, and the highest dose of MTB-VAC, as you can see in the green, did have higher frequencies of CD4 cells through 1 year of age compared to the BCG group. And those doses of vaccine were comparable for 2.5 times 10 to the 5th CFUs for both BCG and MTB-VAC. And notably, there was a dose-related increased frequency of IGRA conversion among MTB-VAC recipients due to cross-reactivity of the vaccine with ESAT6 and CFP10 antigens in the commercial IGRA assays. And that could really limit the use of MTB-VAC for preventing infection or prevention of infection trials, as well as use of that assay for future clinical use. And so to date, a dose-finding phase 2 trial has been completed, and a phase 3 efficacy trial to prevent TB disease among HIV-exposed and unexposed infants just started last month. So moving to adolescents, a recent trial of BCG revaccination in 2018 demonstrated about 48% efficacy for BCG revaccination to prevent sustained QFT conversion among HIV-negative, IGRA-negative adolescents in South Africa. It also showed about 45% efficacy to prevent high magnitude quantiferin levels, which may have clinical significance since this cutoff has been associated with a 30 to 40-fold increased risk of TB disease in children and adults. And an ongoing larger trial among children and age 10 to 18 is underway to validate these findings. So M72, kind of moving from the whole cell vaccines to protein subunit vaccines. M72 is a recombinant protein it includes 2 TB antigens of virulence factors 32A and 39A, and an organic adjuvant AS01E that's given in 2 doses 1 month apart. And a phase 2 trial in South African adolescents with and without TB infection showed that the vaccine is safe without any serious adverse events. Solicited generalized adverse events were more frequent in adolescents with prior MTB sensitization detected by IGRA, and M72 was immunogenic, inducing multifunctional CD4 cells, notably of higher frequencies among adults or among adolescents that were IGRA positive. And an exciting development for the TB field was a phase 2b trial in IGRA-positive adults, which found nearly 50% vaccine efficacy to prevent microbiologically confirmed TB disease. A phase 2 trial, including adolescents, oops, sorry, having trouble with my mouse. A phase 2 trial, including adolescents over 16 years and adults with HIV, is ongoing, and a larger phase 3 trial is being planned right now with Gates MRI, and that is anticipated to also include adolescents. So this is just an overview of the current pipeline of pediatric TB vaccines. A few of the candidates that don't have published data in children yet are the ID93 and the ChAdOx 85A vaccines. And so the ID93 is a protein subunit vaccine in phase 1 trials that will include that are including adolescents that are ongoing. And the ChAdOx 185A is the MVA is the kind of the chimp version of the 85A adenovirus virus vectored vaccine. So those are coming up in the pipeline. And then you can see, there's just overall more candidates that are being tested in adolescents compared to infants. I wanted to actually highlight a bit of, just the TB vaccine pipeline and people living with HIV. In addition to, we're focusing overall on children, but just to highlight that TB is still the leading cause of mortality in both adults and children with HIV. And there's still high risk despite ART and high CD4 counts. So this graph that I'm showing is among adults, but there's a study published in children as well, just showing that overall we still have a higher risk of TB despite our antiretroviral therapy. And so we still need to be thinking about trials in adults and children with HIV. And so I quickly wanted to highlight one of the studies that is launching that many of us at Emory are involved in. So this is called the LEAP trial. And so it's supported by Impact 20. It's the 2035 study of Impact and HVTN networks, HVTN 604. And it's a phase 1-2 study of the safety and immunogenicity of VPM compared to BCG revaccination. And we're really focusing on this pre-adolescent timeframe of children ages 8 to 14. So I'm the protocol chair working with Cheryl Day, who's protocol vice chair, and we have Jyoti Ranjarakan as well here as one of our co-investigators. So it's been really fun to work all together with other international investigators on this trial. And this trial really evaluates the safety of VPM vaccination and BCG revaccination in children with HIV, with and without HIV. And then we're looking at just differences in immunogenicity as well, focusing on cellular immunogenicity, but we'll also have secondary endpoints looking at humoral immunogenicity. And overall, this will be in South Africa among children who received the BCG vaccine at birth, and then between 8 and 14 years of age, they'll be randomized to receive VPM, BCG, or placebo. And they will be following them for 48 weeks on study. And we're including, about 9 sites. So a combination of Impact affiliated sites as well as HVTN affiliated sites. A few things just to kind of put this trial in context is that, with the very, high concerns of safety and immunogenicity in the pre-ART era for people living with HIV, there really was a focus on only using subunit and killed whole cell vaccines for people living with HIV. And so, the M72 candidate has been evaluated in adults with HIV, finding similar immunogenicity for adults with HIV on ART, but much lower immunogenicity among adults who are not on ART. And there is a larger trial specifically just testing M72 in adults, adolescents and adults with HIV going down to age 16. But, because we don't really know what is the vaccine efficacy going to be in younger ages, in the population with HIV and how scalable this vaccine is going to be, we are wanting to expand options for other vaccine candidates in people living with HIV. One of the other candidates, the DAR 901, is a killed vaccine that was tested in a larger prevention of infection trial in Tanzania, and there was no safety signal, although this trial was a bit underpowered for that signal of prevention of infection. So just to plug that we really do need to expand vaccine candidates in people living with HIV. And when in the beginning, when we were discussing our trial, it was really, there was a lot of resistance about actually testing live wholesale vaccines in people living with HIV, even who are on ART. And so this is the first trial to really evaluate a live TB vaccine in kids with ART. And also, so that comparison of IGRA positive and IGRA negative is also going to be important to really understand how much that modifies the immunogenicity. And part of in the development of that trial, we established relationships with across the networks. And so recently just wanted to highlight a publication that we were able to be co-authored on, was just looking at developing our whole roadmap for developing TB vaccines for people with HIV, since it's a priority population. And so that's been a neat process to go through. So kind of the next step that I just wanted to highlight is what is our uphill climb and what are the unique challenges that we face with TB vaccine development. So overall, TB vaccine trials, in the older time periods, really started with uninfected young infants and would follow them for many, many years, through infection and then until disease, presented. We've been able to, shorten trials by introducing the, prevention of TB infection as an endpoint, but it's still unclear if preventing TB infection really will correlate with TB disease prevention. And so it is a helpful, but albeit imperfect endpoint for our studies. And MTB is also just a very complex pathogen. So while COVID, for example, has only 30 genes, TB has around 4,000 genes, over twice the amount of Haemophilus influenza, which is another bacterial vaccine preventable pathogen. So when we think about TB, compared to COVID-2, COVID-2 had a clear antigenic target that was responsible for viral entry into the host cells. But TB, on the other hand, has a very dynamic lipid membrane. There's distinct composition that's stage specific during active bacterial replication or latency. And TB has really evolved to have multiple host evasion mechanisms that make its antigenic vaccine targets pretty elusive. So the central challenge, to the TB vaccine field is that we don't have immune correlates of TB protection. And our current measures of immunogenicity don't equal protection. So that main concept that immunogenicity is not protection. This was an older study in 2010, but it looked at, following South African infants who had received BCG vaccine at 10 weeks and comparing their immune responses at that early time point among cases and controls, cases with TB who later developed TB after 2 years of follow-up, those who had household controls who were TB exposed but also did not develop TB, and then community controls as well, matched by age. There was similar frequency of this measure of immunogenicity of Th1 cytokine positive CD4 cells, which is a common measure of immunogenicity in our studies. And so it just highlights that, we really need an immune correlate of protection. And this challenge of not having an immune correlate of protection was really highlighted when the first large TB vaccine efficacy trial of MVA-85A had really disappointing results. And this trial was conducted among nearly 3,000 South African infants and elicited, robust interferon gamma responses by ELISpot and robust polyfunctional Th1 and Th17 immune responses, but there wasn't any efficacy to prevent either TB infection or disease. So the search for TB immune correlates of protection is ongoing in non-human primate challenge models. They've demonstrated that IV BCG vaccination was highly protective after MTB challenge compared to intradermal and aerosol administration. And that's led to further characterization of non-conventional immune markers outside of CD4 responses that may really correlate with protection, including both humoral responses, including IgM, and glycolipid-specific T cells. And currently, ongoing studies are investigating a wide array of potential correlates of protection. And so there's, both in the non-human primate models as well as in human trials using stored samples within the large BCG revaccination trials, looking for correlates of protection from MTB infection, and within the M72 trials, looking for correlates of protection from TB disease, and really just expanding to kind of the entire kitchen sink of immunologic approaches as well as types of cells and parameters. So in terms of like thinking about where we're going and how in the TB vaccine field, how can we learn from this recent from the COVID pandemic to move towards a tipping point in TB vaccines? I kind of had a few lessons of learning points. So the first lesson is really to name the problem. So the WHO declared COVID a pandemic only a few months after it was discovered. And after that, a flurry of infographics have really emerged to compare coronavirus pandemic into all the most deadly pandemics in the history over time, and the Black Death is commonly hailed as the deadliest plague of all time. But what's consistently missing from this picture is tuberculosis, and so TB is an unnamed pandemic. It was the first documented, as I mentioned earlier, in human skeletal remains as early as 8,000 BC. And while we don't have precise mortality estimates, over 10,000 years, it's caused more human death than any other pathogen. And TB is a pandemic, and we really need to name it as a pandemic to fuel global action. The second learning lesson is really to think about adopting a moonshot mindset. So JFK challenged us to put a man on the moon in a decade, and we made that goal. Operation Warp Speed aimed to deliver over 300 million doses of a safe and effective COVID vaccine by January 21st, 2021. And we did it. We were able to get a vaccine. And how did we do that? To quickly deliver a COVID vaccine, the US government invested $18 billion upfront into 3 different vaccine platforms. Each platform had 2 to 3 manufacturers per platform. And this really allowed lower risk to manufacturers at the start and allowed for a healthy competition to really race to the delivery of a COVID vaccine. We need to apply these principles to TB vaccine development, which will require large-scale global investment, expansion of vaccine platforms to include RNA and DNA vaccines, and creating a heightened capacity for both global manufacturing and enhanced market demand. And colleagues have recently published a global roadmap for TB vaccine development. And what we need now is really investment and a race to spur on towards this moonshot goal of accomplishing or achieving a new TB vaccine. One of the aspects of accomplishing that huge goal was really, streamlining systems, and COVID really taught us a lot of how streamlining systems can really make the vaccine development process faster, including adaptive clinical trial design with overlapping phases, master protocols, and flexibility for ongoing case detection, and working towards more expedited regulatory review and pre-planning manufacturing and distribution. And lastly, what we learned from COVID is that, COVID taught us that we really have to prioritize the public health impact. So as Walter Ornstein says, vaccines don't save lives, but vaccinations save lives. And as we perform TB clinical trials, we really need to be working with communities to identify parameters of vaccine acceptability and identify areas of hesitancy ahead of time. And we need to be working on shrinking the global equity gaps by enabling widespread access of an affordable vaccine that's broadly deployed. So, the 3 lessons that I think we all should remember is naming the problem, shooting for the moon, prioritizing impact, because it is time to end tuberculosis. So just wanted to thank all my mentors and collaborators and funders. I'm so excited for this track to be starting and to have enhanced collaboration with UGA and Emory and others around, and just open it up to any questions now.
Thanks, Lisa. We don't have the chat function working, unfortunately, so I am just going to invite people to open up their mics maybe raise your hand and then we'll just call on your name. Sorry. Usually the chat makes it a little bit more efficient, but we have it disabled for some reason, I don't know why. So who wants to go first? Anyone, you can use the hand raise or just open your mic and ask. I see Chris wants to go first. Dr. Wayne, please go ahead.
Yeah, Lisa, thanks so much for that nice talk. So much going on. We had a vaccine initiative in the very first TBRU in 1994 and 95, and it just seemed like light years away that we would ever get anything close to a real vaccine. But a lot has been done, a lot has been accomplished. So, and I'm glad you're in the midst of it all. I have a question, and it's puzzled me in some of the vaccine discussions, and that relates to how do you know whether you're preventing infection or not?
Yeah, I mean, that's the challenge. We have imperfect measures of infection, right? So we have no way to really detect the bacillus itself. And so we're just measuring immune responses. And those are imperfectly measured. One of the, we currently just look at the interferon gamma, released by CD4 and CD8 cells. That's what the commercial IGRA assays measure, and they're imperfect. Well, in younger children too, they're less sensitive, and we don't have as robust data in the youngest children under 2. So yes, we need a better marker for the actual bacteria, but we don't have it yet.
So whatever vaccine we develop, it actually can't or shouldn't stimulate an interferon gamma response, right?
Well, I mean, I guess that's the question. I mean, hopefully
we wouldn't be able to distinguish it from, we wouldn't be able to show in a clinical trial that it's a new, that there's a new infection that has occurred.
Yeah, so like an MTB vac, it does cross-react with the IGRA assays, and so you can't really do a prevention of infection trial for MTB vac. And so right now their trial is going to be an efficacy for TB disease as the endpoint. So yeah, that's very true. I think they are working on getting an assay, similar to the IGRA, but that doesn't have cross-reactive antigens. But I believe that that's been difficult to develop. So I think we are maybe limited in that aspect for MTB vac.
And I don't know whether anyone else has a question they're asking, but this is burning in the back of my mind, so I'm going to go ahead and ask it. What's the immunologic mechanism for preventing infection?
Well, that's what it's so interesting. We don't know yet. My K23, we're looking at antibody responses, and I'm very, I think there's a lot of exploration in mucosal immunity. So we don't know, but the non-human primate study that I mentioned did show some signal in terms of IgM, and there's some other basic science papers, looking at IgA as well. So I do wonder if there might be an antibody role in that, but it's probably multifactorial given the complexity of the pathogen. And we certainly know that, adaptive T cell responses are really important, but whether that's important in the infection prevention process versus the progression from infection to disease, is yet to be determined.
I mean, I always think of that what protects us from infection would be innate immunity, which, as I understand it, a vaccine won't do anything to it. So there must be some hypothesis of this intermediary time between innate immunity and adaptive immunity that, the adaptive immunity doesn't show up, but somehow the vaccine will block the growth of the organism. Anyway, it's such a complex thing about understanding that initial infection with MTB, and that's been a challenge. All right, great. That wonderful presentation gives me hope.
I see Christine Nelson has her hand up.
Yeah, thanks, Julie. And thanks, Lisa, for the talk. That was great. Kind of piggybacking on Chris's question about mechanisms of protection. One of the things that has always kind of puzzled me about both the M72 results and the BCG revaccination results for the trials that were done, the phase 2B trial done recently, is that the survival curves, like you don't see much effect early on. Like the effect that you see is mostly quite a bit later on in the follow-up period. And I would contrast that with like COVID-19 vaccines, for example, where actually the greatest effect that you see is in those few first weeks after vaccination. So yeah, I wondered if you could speak a little bit to how that, if it does relate to kind of the mechanism of protection for these vaccines.
That's a great question. Yeah, I mean, I think it probably does. I think you raise a good point. And I think we don't know at the end of the day, but I do think that is a very good point, and it might point to the importance of adaptive immunity as well. Then, given the time frame.
Yeah. Okay, fair enough. Thank you.
But I don't know.
Maybe there is a unifying theory, immunological theory.
I think Cheryl's out of town, unfortunately. I have a few other immunologists. I think Ankita Garg is on the call. I know that you're an immunologist. I don't know if you wanted to pipe in.
I'm sorry, I just missed the question. What was the discussion about? Yeah.
Oh, it's okay. No worries.
I'm sorry. Yeah, no.
Okay. I think none of us really know.
Okay. Any other questions, comments? I see John, I think. Go ahead, please.
Hi, thank you. This is John Jareb over at the TB division. We saw a huge swell of interest in the possibility of BCG preventing COVID-19 or reducing mortality or something, and a lot of articles, a lot of editorials, and ultimately it looked like it probably isn't worth much more attention. But it also returned our awareness of the so-called off-target effects of BCG with infant mortality from several causes probably being decreased by BCG at birth. So I'm wondering if the current trials have the bandwidth or the design to capture any of those so-called off-target effects of vaccination. Over.
Yes. So yeah, that was seen, for example, in the BCG revac trial that was published in 2018, that they did see lower rates of upper respiratory infection. So kind of another signal about the off-target effects. I mean, certainly for mortality as an endpoint, you need massive trials. But in terms of, looking at URIs and COVID as an outcome, that's certainly being pursued. And then, in terms of like the actual immunological mechanisms of, looking at the, epigenetic reprogramming and the immune training that is, potentially responsible for that, samples are being collected so that can be evaluated. But, in terms of, published findings still from that, or even knowing exactly what we're going to measure, I think is still to be determined. But I think we're all interested in learning more about that.
Ankita.
Yes. Hi, Lisa. Yeah, that's interesting. And I'm sorry, I just missed that discussion. But I have a question in continuation with the sample you said being collected to look into the epigenetic modifications on BCG and versus its effect on COVID. But are you also considering other infection samples? Because epigenetic modifications not only happen with BCG, but in any of the yellow fever virus infection with all of those pathogen also. So are those also being considered for epigenetic changes, or it's just BCG?
Well, I think the question was about, the trials, the TB vaccine trials. So yeah, I mean, I think they're being looked at for TB vaccine trials. I can't really speak to the other vaccine trials and how much they're looking at that, because I'm not involved in non-TB vaccine trials. But certainly, I think it's a really exciting area that I think we're learning a lot more about in terms of what's driving that: vaccines, infections, HIV infection driving epigenetic changes, other parameters. So,
okay, thank you. But I have a question. It's just not written, because in one of the slides you mentioned that the hesitancy for the vaccination should be overcome. So in field, do you often experience hesitancy for the BCG vaccine? I know there are regions where there is hesitancy for other vaccine intakes, but do you experience that for BCG vaccine also?
Sorry, did you mean vaccine hesitancy, or did you mean
hesitancy, like people are resistant to take BCG vaccine in certain geographical locations?
Yeah, no, it definitely can vary by region, but it is, I think part of the pragmatic public health of it just being given at birth for children who are born in the hospital. I think pragmatically that ends up being the reason why it's the number one vaccine administered globally. So I think that's a kind of a structural reason, and it has I don't believe there's been studies over time looking at, because vaccine hesitancy certainly has increased over time, but I don't think that there's been studies looking at particular acceptability or hesitancy around BCG. But certainly that is something that we are nesting questions in, like for the current trial that we're running in the South African pre-adolescents, really getting some acceptability data there, because there actually was a really old study. I mean, I'm not that old, but like in the 1980s, they were doing some revaccination work in English adolescents, and there was differences by sex, like males and females preferred different things about the vaccine and wanted the vaccine in different places. And so it's as COVID had shown us, we really need to be thinking about that now before we come up with a wonderful vaccine that we're excited about and that nobody actually receives. So yeah, it's an important area for sure.
Okay.
Well, I'll just chime in that BCG actually has a very checkered record in terms of its safety. There's a famous episode in Germany where BCG was given to children, and unbeknownst to the people in this, I don't know whether it's an institution or what, I don't remember. They actually gave live M tuberculosis to the children, and 260 kids got infected, were treated, and 2/3 of them got TB, and many of them died. And this was almost 100 years ago. I think it was 1923 that it occurred, or thereabouts, in Lübeck, Germany. So, and that incident stayed in the minds of people all around the world up until after World War II. So it wasn't the vaccine that was dangerous per se, it was how it was implemented or how it was given. And that has some resemblance to the COVID vaccine, how that was ultimately rolled out and given and how it was accepted. But that Lübeck disaster really put a black eye on BCG for decades.
Any other comments? All right, so if there are no other questions or comments, let's just put our hands together, the icon hands, and thank Dr. Cranmer for such a wonderful talk today. And thank you everyone for joining the Global Health Seminar, and please come again. We have our next one on 1st December, which is World AIDS Day. And so we'll have our last one in the fall series on that day. Thank you again, and have a good rest of your evening. Thank you.